Today we will talk about how many proteins work together in the DNA replication and repair. And to that I'm going to start talking about history, about how when we start studying the process of replication there were three models, and now we know that the replication is semi-conservative. Okay, let's suppose that here you read semi-conservative. Second thing that we will talk today is about the origin of the replication and how they differ when they are prokaryotes, when they are eukaryotes, when you have a big genome. Then I will talk about the leading and the lagging strands for DNA replication. Then I will talk about all the different proteins that are involved in the DNA replication. Later I will move to how the DNA proof and repair the DNA. And finally, at the end, I will talk about how the DNA starts getting smaller and why this happened and how the cells try to avoid that this happen. Because if the DNA start getting smaller and smaller, what is going to happen is that you're going to start losing genes, and this is one of the reasons why cancer can develop. So let's start with the first one. So possibly you know this is the structure of the DNA, and you right away you think about DNA when you see this structure. However, if you took genetic revolution, you know that this is a long history to be able to achieve this. And now I'm going to tell you the next step, what happened here, how we start studying the replication models. And to start, I want to just show you a video of the DNA double helix.
DNA consists of two complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.
Here in blue of carbon, and here they go from 5' to 3', and here they go from 2' to 5', so they're antiparallel. And this is very important because here you have hydrogen bonds and all these important details about the structure of the DNA were important to understand how the DNA replicates. So when Watson and Crick start studying the structure of the DNA and they proposed the model, they found that the structure and the function of DNA is manifest on the double helix. And not only that these two change, that you have one chain and the other chain will be maybe important to understand how the DNA replicate. And this is not an idea that hopefully they copied from Rosalind Franklin. This actually was an idea that it came from them. So remember, Watson and Crick note that the base pairing suggested a possible coping mechanism of this genetic material. In this slide, I want to show you that the basic principle of base pairing was important because it helped to make a template strand. So let me show you. First thing that I want that you remember is that now we know that the replication is semi-conservative. That was the model that was correct. And I will show you this diagram to try to explain what is a semi-conservative replication. Here you have two change, and the two change are the parent strains. And then you apply heat or you use an enzyme to separate these two change. And then as a result, you have an all parent strand here and a parent strand here, but here you have a sister or a new strand. And how you can make it, because these are complementary. So let's summarize here. I want that you remember that since the DNA strands are complementary, they can act as a template for building a new strand during replication. So the second thing that I want that you remember, that in the DNA replication, the parent molecules unwind, as I show you here. In the case of DNA in living organisms, they do it using enzymes. However, in the lab, we do it just by heating and breaking these weak bonds that are the hydrogen bonds. And finally, the third thing that I want that you remember is that these new daughter strands are built on base pairing rules. And so this is the semi-conservative model of replication. Here you have a parent strand. Here you have a parent strand. This dark blue is a parent strand, and here you will have a parent strand. While the other ones are new strands, new sister strands. But as you can see, every time you will start getting new strands. Here you only get half and half. Well, here you have nothing, and here you have half and half. And this was actually an experiment that it was done that could be explained by the model proposed by Watson and Crick. And we are not going to get into a lot of detail about that experiment, but I want that you remember that the model of Watson and Crick, the semi-conservative model of replication, predict that when a double helix replicate, each daughter cell will have one parental strand and one newly strand. And this was proven experimentally. So there are three alternative models of replication. So as I told you, the semi-conservative was the correct one. However, in the beginning, we thought that it could be a conservative model. And as you can see here, you have apparent strands. And here you will have or a new one or an old one. And then this will produce only new ones here. And only one old one that look like the parents. Another potential model was the dispersive model, that instead of making only entire copies of the one strand, they were proposing that they were pieces of copies. So here you have each strand is a mix of a new one and an old one. So in this case, you just have mix of the parent plus the sister strand. So okay, so now I want that you remember from this slide that there is something called a conservative model, that there are two parent strands that rejoin, and also that there is a dispersive model. And then in the dispersive model, each strand mix an old and a new strand in one single strand. So here, a new strand plus an old strand plus a new strand. And as I said before, here the correct one, although there were three models, the correct one was the semi-conservative. Well, that was the correct one. So now let's go to the origins of replication. And now I want that you watch this video of your textbook that explains what is an origin of replication.
DNA replication, an overview. In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules. Here's a simplified close-up of replication showing what happens along one of the template DNA strands. Using a supply of free nucleotides, DNA polymerase assembles a new DNA strand along the template strand, following the base pairing rules: adenine pairs with thymine and cytosine pairs with guanine. Now let's look at the DNA replication process. Replication of a DNA molecule begins at a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules. In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we've untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical daughter DNA molecules. Now, let's review the synthesis of DNA by assembling a new DNA strand. We'll use the top DNA strand as the template and choose from the free nucleotides below. Note that D stands for deoxyribose and R stands for ribose. We're only using the deoxyribose nucleotides because we're building DNA, deoxyribonucleic acid.
Now I'm going to talk about the origins of replication, and as we watch in the video, here I'm going to talk about the origin of replication using this diagram that talks about E. coli. It's a bacteria and it's a much simpler organism than humans, so we can study the basic principles. To that, I want to tell you that the replication, both in bacteria as well as in eukaryotic organisms, display an incredible speed and accuracy. On how this is done, because there is a complex of proteins that work together to achieve this. And the first thing that I want that you see in this diagram is that they show you that in E. coli you only have one origin of replication, and from that spot you start dividing the DNA, and now you start copying the DNA, and now you have a new strand, and you form two things. One thing that is called a replication fork, that is the region where the DNA separates, and also this entire structure is called a replication bubble. So the replication bubble grows. In this section of the diagram, you can see that most of the DNA is copied, and at the end you end up with two daughter sets. So to recap, copying the DNA is remarkable in not only in its speed but also in its accuracy. Also, you have dozens of enzymes working together in this process of DNA replication. Then I talk about the origin of replication, that is, the replication begins at a particular site called origin of replication. And finally, I told you here that the DNA strands are separating, opening up in a bubble. That is called a replication bubble. Now let's talk about the eukaryotic chromosomes, where you don't have one, but you have multiple origins of replication. So as I told you before in the previous slide that the eukaryotic were much simpler, then when we moved to study eukaryotic organisms, we found that there is no only a single point, not a single origin of replication, but you have multiple origins of replication. And I wonder, you think, why? Why you are going to have only one in a bacteria, but you need multiple in humans? So stop at the video and try to think about it. Okay, maybe you already have the answer, and the answer is because the DNA of the bacteria is very small. If we have only one origin of replication of human, copying a single cell will take forever. And this is why we need to have to make the process very fast, we need to have multiple origins of replication. However, you still keep the same principles. So if you have here several origins of replication, but you have a replication bubble here. Here you can see the replication fork. And when you are done finishing each replication bubble, you can have two daughter DNA molecules. So to recap, a eukaryotic chromosome may have hundreds or thousands of origins of replication. Replication proceeds in both directions, from the origin until the entire molecule is copied. Now I'm going to talk about the leading and the lagging strand. To start, I want that you watch this video of your textbook that summarizes this important topic.
First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together.
Now I'm going to talk about the antiparallel structure of the DNA, and this antiparallel structure that you already studied affect the elongation. So here we have a diagram that shows the structure of DNA, and I know you remember that here we have a 5' and here you'll have a 3'. So it is going this direction, the backbone between sugar and phosphate going this direction, and here it goes in the opposite direction. However, this seems to not be related to the DNA replication. However, when the DNA open and you have one strand here and the other one is a strand here, so imagine that you start creating a copy of that. Now one will go in one direction and the copy will go in the other direction. So this is why this structure that is antiparallel will produce difference in the DNA as you're watching the video, and it will create these two type of strands that are lagging and also a leading strand. So the antiparallel structure of the double helix affect the replication as I just showed you. And we just remind you that the DNA backbone has 3' and 5' carbons, and they go in opposite direction. And this is why they are called antiparallel. However, when there is a new strand, only can elongate in the 5' to a 3' direction. So the enzyme that is in charge of this process only goes in one direction. So when you copy this DNA, it will only go in one direction and the other one will go in the opposite direction, creating a very interesting scenario that we will talk in more detail in a second. Now I'm going to talk about the leading strand to start understanding the synthesis of DNA. And here in this diagram I want to show you how the leading strand start being synthesized. Here you start with an origin of replication, and the DNA polymerase locate here and start moving continuously toward the fork of replication. Same thing happened here. It goes to the origin of replication and it goes synthesize the DNA continuously towards the fork of replication. So first thing that I want that you remember is that you have one template strand, and using that template strand, the DNA polymerase located in that section and start synthesizing the DNA continuously, and it moves toward the replication fork. Now I'm going to start talking about the lagging strand that require different mechanisms. So the first thing that I want that you see here is that, well, the origin of replication is here. In the leading strand, you go toward the fork of replication. Here you go in the opposite direction. You go away from the fork of replication. And as a result, you have to do it discontinuously. So you create these short segments that are called Okazaki fragments. And the Okazaki fragments are joined later together by this enzyme called ligase. So to summarize, to elongate the other strand or the lagging strand, first the DNA polymerase synthesizes discontinuously. So this is the first thing that I want that you remember from the lagging strand. The second thing that I want that you remember is that the DNA polymerase must work in a direction away from the replication fork. And finally, third thing, that the lagging strand is synthesized as a series of small fragments called Okazaki fragments, which later on are joined by the DNA ligase. And as we watch in the video, it takes longer. This is why it's called lagging strand. So we already talked about the models of the DNA replication, and we talk about, say that this is semi-conservative. We already talked about that in bacteria there is one origin of replication, while in eukaryote you have multiple origins of replication because they are much complex. Later we talk about that the leading strand has a different characteristic of the lagging strand, that this one goes fast while the lagging strand goes discontinuously, so it's much slower. And now we're going to talk about the different proteins involved in the DNA replication. In this slide, I'm going to talk about the DNA replication complex. And the DNA replication complex is all the proteins that are working together to replicate the DNA. And there are many. And similar to the origin of replication, we are studying prokaryotes to later studying eukaryotes. Why? Because the prokaryotes are much simpler. So here I'm going to show you some of the enzymes that are used in bacteria because they are simpler. In eukaryotes, again, the same principles are used, but instead of having, for example, here you have two polymerase. One that is in charge of synthesize the DNA, the new template, and the second one that is in charge of remove the RNA during the replication. In eukaryotes, you will have much more version of these enzymes and all ones that are more specialized. However, the same processes are happening. So for example, eukaryotics also have helicase that allow to separate the DNA. You have single strand binding proteins that allow to stabilize when you separate the DNA. When you have these enzymes here, the DNA is not able to attach so easily. You also have the topoisomerase that are very important because it removes the mechanical stress. So when the DNA start folding and bending in an atypical way because there is a bubble of replication, these enzymes allow to make small cuts, so the tension is released and we don't break the DNA and lose some portions. We also have this very interesting enzyme that is a primase that adds an RNA fragment. And why these RNA fragments are important? Because if you have an RNA fragment, so you have an RNA fragment, then this enzyme can start attaching here and start synthesizing DNA. And finally, the DNA ligase is in charge of joining fragments of DNA that they were separated. And now that I talk a lot, I want that you do a small assignment. Stop the video, and I want that you match these two columns. You don't have to upload this into on Moodle. Don't do it. There's no need. I just want that you match the name of the protein. Here you have the name, and here you have the function. So what I want that you do is that you go and say, okay, what is the function of the helicase? You can go back into the video. You can go in look in your textbook. You can go and watch a video, or you can just Google it. What is the function of the helicase? Maybe it's join the Okazaki fragments? I don't think so. Remove the RNA? Don't think so. So you can go and start checking all of them. And in the next slides, I will give you the answer. So stop the video and try to do this small assignment. So here is the answer key. 1 goes with C. 1 and C are together. So the helicase is the one enzyme that unwinds the parental double helix. And you can keep reading and make sure that you did everything right. If you have any questions, let me know. Now I want that you see another video of the DNA replication, and this review all the topics that we just talked about: the template strand, the lagging strand, and the different enzymes.
As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces.
Now I'm going to give you a small summary that you can find in your textbook of the bacterial DNA replication. So first step, you take the helicase. Second step, this will separate the DNA. And the second step is stabilizing the DNA. How you do it with this single strand binding proteins. Here in this schematic, they said to you, you go to the leading strand, synthesizing continuously. They are missing in this diagram the 4 and the 5 steps. That is the primer, and then the polymerase tree. But I think they are just trying to make it simpler. However, in the ligand strand, again, you need the primase that allow to set the RNA here. Then, if you remember, Later you go to the polymerase tree that synthesizes the DNA again in the moving away from the fork of replication. Then the polymerase tree remove the primer, and to finish, the ligase is going to join the two fragments of DNA that they were separated. Okay, so we finish with this section. So we just explained the current model of DNA replication, and as I said before, this is from bacteria, but the one from eukaryotic is, although it's similar, has much more proteins and is a little bit more complicated. Now, we're going to talk about how the DNA makes sure that it did a good job and what happened, how the mechanisms that repair DNA. So in a similar way that when we are writing a document, a Word document, there is a software that allows us to correct the mistakes. The same thing happened with the DNA, and there is a group, for example, the DNA polymerase, proofread the DNA and make sure that all the sequence of nucleotides are correct. There are also enzymes that are doing that constantly, that are checking that the base pair are properly attached. And why we have to correct these things? Because, for example, chemicals like this one that can produce mutations, and there are, for example, radiation, free radicals. There are so many examples of smoking, of chemicals that can produce mutation. So the DNA polymerase proofread the newly made DNA, replacing any incorrect nucleotides. And this is the first thing that I wanted you to remember. Second thing that I wanted you to remember is that mismatch repair of DNA. Enzymes correct the errors in base pairing. And the third thing that I told you is that the DNA can be damaged by exposure to harmful chemicals like this Roundup. It's a perfect example. Or physical agents that can undergo to spontaneous changes. In your textbook, they show you this nucleotide excision repair of DNA damage. So let me show you what is happening in this diagram that your textbook is showing. So they show that, for example, here, all these sections are properly attached to each other. There's the correct base pair. However, here, if you see, there is a mismatch. So a nuclease is going to cut this section and remove it. And then a DNA polymerase is going to come here and attach the right sequence. And then the DNA ligase is going to join these two fragments of DNA. So in a nucleotide excision repair, a nuclease cuts out and replaces the DNA damage stretch of the DNA and replace the damaged stretches of DNA. Now let's talk about the implication of the DNA repair. And I wanted to tell you something. Although the DNA has a very efficient machinery to repair the DNA, they're very good at doing that. It's 99 point something. Yeah, it's very close to 100%. However, those small percentage will allow that there are mutations, and this will have an effect on the living organisms. Why? Because they can pass from one generation to the next one, some of those very few mutations, and these very few mutations, in most of the cases, most of them died, and few of them, they will be advantageous for the organism, and this will produce the diversity of life. So for example, here you have this diversity in felines. You see the colorations, and some of them, they could be good and some of they could be bad depending on the environment. So if you live, for example, in a place where there is snow, this could be useful. However, in other places, if there is no snow, this will be more useful. So remember, three things to recap. The error rate after proofreading is low, but it's not zero. And as a result of that, the second thing that I wanted you to remember is that the sequence changes may become permanent and can be passed to the next generation. And as a result of that, these changes, these mutations, are the source of genetic variation upon which natural selection operates. To finish, we already talked about the semi-conservative replication. We talk about the comparison between prokaryotes and eukaryotes. We talk about that the leading strand goes continuously and the lagging strand goes slowly. We talk about all this protein complex and how the DNA use nucleases to repair, and there is more information in your textbook to read. Finally, we're going to talk about telomeres, and we will talk about this in more detail in the classroom. Your chromosomes, one of the reasons why we age is because our chromosomes will start getting smaller. And this is a chromosome. If you remember, at the end of the chromosome, you will have these structures that are called telomeres. And as every time that the cell divide, and divide, the telomeres, these sections of the chromosome that have repetitive sequence, here you will have genes. Here you are just going to have repetitive sequence that don't produce proteins. So no protein. It's not, you will not have protein there. They will be lost. And here there will be no problem, no problem, no problem. However, when your cells start getting old, you will start losing all of the chromosomes that actually have gene, and this can be problematic. So we're going to talk about that. From this slide, I want that you remember that your cells go to repeated rounds of replication. Every time that your cell divide will have to replicate and produce a shorter DNA molecule with an even ends. And this could be dangerous in the long term. Now I'm going to talk about these structures in your chromosomes that are the replicating ends of the DNA molecules. Here you have a technique, these are real chromosomes, and they stain it to show the repetitive sequence of DNA that don't code for proteins. And if you see at the end of the chromosomes and in the middle, you have this sequence of DNA that are just repetitive, that they don't produce proteins. Here you have a diagram that shows the telomeres, these structures that are at the end of eukaryotic chromosomes that are very big. So here you are going to generally have more proportion of genes, while here you have repetitive sequence that they can be lost and there is no change in your cells. So remember, in eukaryotic cells, the chromosomal DNA molecule have a special nucleotide sequence at their ends called telomeres. And those telomeres do not prevent the shortening of DNA molecules, but they postpone the erosion of genes near to the end of the chromosome. And it has been proposed that the shortening of the telomere is connected to aging. Okay, now that I told you how we are losing the telomeres, how they are getting shorter, now I want you to think about what about if we prevent aging by making big telomeres? Yeah, what about if we make just big telomeres? And actually, life already planned that, and there are organisms that do that. For example, in the case of humans, our germ cells, they have an enzyme called telomerase that make the telomere longer. So in that way, we don't lose our gametes full of mutations, or they are not too small, and in that way we can continue being alive generation over generation. However, they are not active in all cells. And could you think why they are not active in all cells? What could be the negative effect of have the telomere active in all cells? The problem is that, for example, if you have a telomere, every time that telomeres that are very long, every time that you will have a cycle of cell division, you start accumulating a small mutation. And then you start getting more and more and more mutations. And at the end, when you don't have the telomeres, this will lead to, in many cases, to cell death. If you have long telomeres, you will allow that the cell continue being alive. So in the long term, this can produce cancer. So we have in all our cells the enzyme telomerase, but it's not active. In cancers, on the other hand, you might have the telomerase active, so those cells have very long telomeres and never die. So first thing that I told you is that if the chromosome of the germ cells becomes shorter in every cycle, essential genes will be eventually missing from the gamete that they produce. So what we do to prevent that? We have active the enzyme telomerase in the germ cells. And this enzyme called telomerase allowed to get longer the telomeres in germ cells. However, also the telomerase is active in cancerous cells and allow the cells to never die. So this is why it's only active in few cells in adults. And I told you that there is a relationship between telomeres and cancer. So shortening of telomeres might protect cells from cancerous growth by limiting the number of cell divisions. So if the cells have too many cell divisions, they will accumulate a lot of mutations. So do not have telomeres can allow us to get rid of the very old cells. And also, there is evidence that telomerase activity have been found in cancer cells, which might allow cancer cells to persist. So in class, we will talk more about this. But right now, if you have any question or any doubt, please go to your textbook, go and rewatch the movies, and later you can send me the question by email or we can solve it in class. So have a good day, and I hope that you enjoyed this class.